I will give you a brief guidelines for the final. Okay, the final is going to be mostly but not exclusively application questions.
Of everything?
It's over everything.
Everything.
Everything that you were required to learn from every chapter.
Okay,
What do you mean by?
Is it on the slides or is it on the book?
The book.
The
Oh,
book, everything?
what is?
What do you mean by application question?
I have a lot of questions in here that are application questions. Let's see. Let's see if I can find an application question here in this list of questions. Oh, great. I've hidden them. I can unhide them. We went over these, I believe. Again, idiot for trying to do this without my mouse. Now I got control.
Is it kind of like if you have excess potassium ions, what's gonna happen?
Those kinds of things. Okay. Blood sample contains elevated levels of IGFs, which pituitary hormone we can expect to be, this is an application kind of question. Okay? Yeah, the answer is growth hormone, because they, oh wait, I'm looking at them and you're not. I gotta put the slideshow up on the screen. Okay. Still not there. Try again, slideshow. Wait for this. Blood sample contains elevated levels of IGFs. What other pituitary hormone would you expect to be elevated as well? Growth hormone, because that causes some tissues to produce them. What effect would elevated circulating levels of cortisol, a steroid hormone from the adrenal cortex, have on the pituitary secretion of ACTH?
So we inhibit.
If you have more of this, you would have less of that, would be decreased. Application question. When a person's thyroid gland is removed, signs of decreased thyroid hormone concentration do not appear until about 1 week later. Why? It stays in the blood how?
Binding proteins.
Bound to a binding protein. And that would be a so-so application question, not one. Removal of parathyroid glands would result in a decrease in blood concentration of what important mineral? Calcium. Not an application. What symptoms do you expect to see in an individual whose diet lacks iodine? We already did that one, a goiter and
What about the
decreased level. Pardon?
cardiovascular system?
Cardiovascular system,
Questions.
yes, there'd be a lot of problems there. But no, I'm trying to answer this question. Okay, what about the level of thyroid hormones? Yeah, lower. And body temperature would be down, metabolism would be down as a result of decreased thyroid hormones. What effect would increased levels of glucagon have on the amount of glycogen stored in the liver? Application question. If you have more glucagon, what would happen to the amount of glycogen in the liver?
Decrease.
Would go down. Okay? Because this hormone is secreted when blood glucose levels are low. Well, how does the liver respond? Breaking this down to glucose and releasing the glucose, the amount of glycogen there would go down over time. What effect would elevated cortisol have on the levels of glucose in the blood?
Cortisol increases.
It would definitely increase it. Okay? So, chapter 28. Do you have chapter 28 notes? We've had lots of application questions on these questions. I will put up on, after class on Wednesday, I will put up the list that tells you how many questions on each chapter. Then I'll put up The longer the chapter, the more questions there will be. And then I will also put up example questions from old practicals, old finals, application questions. Okay? You will not have any of the questions in the first 5 exams. None of them will be on the final. 0.
Oh, the question that we did.
Every one is brand new. Yes.
You have the answers too? Are you gonna post the answers for the question?
To the example questions, yes. Just like all the other ones. Yeah.
I'm gonna be in class.
Your exam 5 will be in class on Tuesday, your final will be in class on Thursday. Writing class, same time. If you have another final which is taking up more time, you have to tell me ahead of time, so I can make sure you have enough time for the final. Tell me if anybody has, you gotta tell me ahead of time, if anything is gonna conflict with the final. Or exam 5. Okay? I can take care of it. They're not supposed to, but chemistry does. So, learning objectives. List the functions of the reproductive system. Distinguish between those functions that occur in males, females, and both. Describe the process of meiosis, and we have to focus on this because obviously the reproductive system is going to produce some gametes that go through meiosis. Highlighting the change in chromosome number and structure of each after each division. Explain why meiosis is necessary in sexually reproducing organisms. Describe the different events in meiosis that result in genetic variation among gametes. If I don't mention it, ask me, it's not in my slides. Describe the scrotum, and we're gonna now begin the male reproductive system. Describe the scrotum and its role in regulating the temperature of the testes. Describe the structure of the testes, the specialized cells of the testes, and the process of spermatogenesis. You have seen seminiferous tubules, you saw sperm, let's talk about the process. List the ducts of the male reproductive system and explain their functions. Describe the, I'm not going to cover that, but you're gonna look over that in the book. Describe the structure of the penis, seminal vesicles, prostate gland, and oval and urethral glands, and explain their functions. List the hormones that influence the male reproductive system and describe their functions. Demonstrate an understanding of the changes that occur in males during puberty. And then finally, explain the events that occur during the male sex act. That's the end of the male reproductive system. We're now going to begin the female reproductive system. And it's very similar as far as what we're looking at. Name the organs of the female reproductive system and describe their functions. Describe the anatomy and histology of the ovaries. Discuss the development of the oocyte and the follicle. And describe ovulation and fertilization. Describe the structure of the uterine tubes, uterus, vagina, external genitalia, and mammary glands. And most of the structure you got introduced to in lab. Describe the changes that occur in females during puberty. Explain the changes in the ovary and the uterus during the menstrual and ovarian cycles. List the hormones of the female reproductive system and explain their functions and how their secretion is regulated. Explain the events that occur during the female sex act. Describe the events that occur during fertilization of the oocyte and the process of implantation of the embryo. They briefly describe it here. I should actually remove it because we're going to cover it in detail, in much more detail, in chapter 29 than is here in chapter 28. It goes over it. I'm not going to cover it here because we cover it in more detail in 29. Discuss menopause, including the changes that result from it. Describe the major age-related changes in the male and female reproductive system. Which one has the biggest age-related change? Male or female?
Female.
Obviously female, because they reach menopause and hormone levels drop, and males' hormone levels keep virtually constant in the 80s. They decline very little. Okay, big differences as far as that last part. Okay, the human reproductive system produces gametes. Which sexes do that? Both. Enables fertilization. Both. Produces reproductive hormones. Both. Protects and nourishes a new individual. Just female. Okay, most people understand that. So let's go and look at meiosis. This was covered in 189, but needs to be repeated for most students. If you're one of these students that already know it, congratulations, this will be a brief review. But for those of you that don't quite remember it, we're going to cover it. Okay, sign-in sheet is coming around before I forget it. All of your body cells, except a few, red blood cells, osteochondroblasts, the ones that degrade bone, osteoclasts. Okay? Except for those that have no nucleus or they're multinucleate, every cell of your body has 2 copies of every chromosome, one from the mother, one from the father. Okay? And you have 23 pairs. 2 times 23 is 46. The rest of the cells in your body have all 46 chromosomes. Okay? That number is very important. If that number varies just by 1, you have serious changes to the human. Some of the chromosomes, if you have an extra one or a missing one, you don't even survive development. Become known as stillbirth. Okay, meiosis is a cell division that's going to reduce this chromosome number from diploid, 2N, to haploid, so that when you take an N and an N and put them together, a sperm and an oocyte, put them together at fertilization, you restore the diploid number of chromosomes. So meiosis is vital. If it doesn't work right, you end up with abnormal sperm, abnormal oocytes. So we're going to begin with a diploid cell. It is already duplicated its chromosomes. Now, this one has 2 chromosomes, a long one and a short one. Yes?
What kind of cell do we start with though? What kind of cell is it that goes
It's
to the-
called different things in males and female. Spermatogonia for males and oogonia for females. They have different names, but they're cells, obviously in the ovary and the testes, that are going to divide and begin to produce cells that go through meiosis. And meiosis is the same in males and females, except for some slight variations in the female, and we'll talk about that. Start and stop, start and stop. Okay, so we have a cell, spermatogonia or oogonia. It's going to divide and produce a cell called a primary spermatocyte or primary oocyte. Those cells are going to begin meiosis. They're produced by mitosis. They're going to go through meiosis. The first step is to duplicate their chromosomes. This cell has 2 chromosomes. Diploid number is 2. And it has a short one and a long one. We're going to duplicate it so you got 2 copies of each one. Okay? And remember, I was going to have 4 chromosomes. So N is 2, so 2N is 4. So it's going to have a long red one, a long yellow. One from one parent, one from the other parent. A short red and a short yellow. So you had 4 chromosomes. Each is going to be duplicated, and I don't have enough fingers for that. Okay? So we're going to take the 4 chromosomes, we're going to make a copy of each of them. Now, during meiosis, before you start splitting up the chromosomes, in meiosis, those 2 from the mother that are duplicated pair with the 2 from the father that are duplicated, and crossing over occurs between them. They will break and rejoin the chromosomes and shuffle the genes present on the one from the mother and the one from the father. Okay? That generates genetic diversity. So right after duplication, they pair. You have 2 chromosomes, they duplicate, they pair, then they exchange segments. Shuffling, and this is different, where they break and rejoin is different every meiosis for every cell. So you can produce a huge number of chromosomes, different types of chromosomes from one individual. Okay? After you have, remember you got 1 chromosome from the mother, 1 from the father, they're duplicated, they pair, they exchange, And what happens at meiosis one is you have the maternal and paternal ones, from the mother, from the father, line up on the metaphase plate, and randomly, one of the pair goes one direction, one goes the other direction, forming two cells. This is diploid individual. This is a diploid cell because it has 1, 2, 3, 4 chromosomes. When you separate that chromosome and that one, put them in this cell, this chromosome and that one, and put it in this cell, this is haploid. Yes, each chromosome is duplicated, but they only have 2 chromosomes. 1, 2 chromosomes. This is haploid. So meiosis one, the first division of meiosis, is a reductional division that reduces the chromosome number from 4 to 2 in this example. Everything above there is diploid. This is all haploid. They have one copy of each chromosome. Yes,
Is that still meiosis one?
that's called meiosis one. Meiosis has 1, 2 successive divisions without any DNA replication. The DNA replication occurred right here in this step. Okay? At least I think that's before replication. When you line the chromosomes, the maternal and paternal chromosomes, on the metaphase plate, right here in this step, the arrangement is random. You can put the male one over here, the female one over there, or you can put the male on this side and the female on the other, one from that parent, okay? And when you line them up, there are many arrangements for 46 chromosomes. There's 2 to the 23rd power, different arrangements of those pairs of chromosomes. That's a big number, okay? So 2 to the 23rd power is a huge number. I can't remember what it is. Then you've got crossing over on top of it. And one individual can produce an astonishingly large number of different chromosomes, different, genetically different cells. And then you do the same thing in the other parent, and then they fuse. That's why when you have multiple children, you realize how different they are, because they get different. I mean, they're still all the mother's chromosomes and all the father's chromosomes, but the way they're shuffled produces different children, radically different children. The more you have, the more you realize how different they can be. Anyone have multiple children? None of it? Okay, and David, what do you say? You agree they're,
Yeah, they can be different.
yeah, they can be different. Yeah, they really can. You think you have it all solved with the first one, you go, oh, what do I do with the second? Because they're different, they're really different. Okay, so at this point, we have created 2 cells that are diploid, but their chromosomes are still duplicated. In meiosis 2, we line all the chromosomes up, and then we're gonna, now, remember, we got 2 cells. Line all the chromosomes up and separate the duplicated chromosomes. So you end up with 4 haploid products. Everything above there is diploid, haploid, these are haploid all the way through. You end up from 1 diploid cell producing 4 haploid products of meiosis. Okay? These have 2 chromosomes instead of 4. They have 2 different chromosomes. So in a human, we would start out with 46, and then there are 4 cells that each have 23 chromosomes. Okay? You must reduce the chromosome number, so we can take 23 from the mother, 23 from the father, and they form the 46 chromosome individual. The diploid number must be restored. So the key part here is, of course, meiosis 1 in reducing the chromosome number. Okay, I think I beat that to death. Everybody understand why for sexually reproducing organisms, meiosis must be done to form the gametes? If you didn't and made diploid sperm, we'd put 2 diploid sperm and a diploid egg together, and we'd have a tetraploid, 4 copies of every chromosome. Okay? And those individuals would produce tetraploid sperm, and then they would unite, and the chromosome number would keep going up by a factor of 2 every generation. Yes?
Isn't that the more The more the buildings look alike, the more DNA or not?
Okay, appearance is determined by many genes which are shuffled greatly in here. But if an individual has, for example, if there were dark hair genes, and if mother and father both have dark hair, they're gonna look more similar than if there was different genes shared. So it depends on how similar the genes are that are to be sorted. Okay, let's go and look at the male reproductive system. We're going to use meiosis. We're first gonna look at the production of sperm, going from a diploid cell all the way to a mature looking sperm. They gotta go through the epididymis. When they reach the end of the epididymis, they're now functional sperm. When they're released from the seminiferous tubules or in the epididymis, they're not functional. They gotta get to the end of the epididymis because there's some more maturation that occurs. So we're going to look at going for all of sperm development. You begin with a cell that's gonna develop a sperm till you got morphologically looking sperm in the seminiferous tubules. As you know, sperm development, spermatogenesis, occurs in the seminiferous tubules. So, first thing you have to realize is these sperm are going to develop in seminiferous tubules. There's a basement membrane up here, and there are these large nurse cells that are shown by these areas of color. Those are nurse or sustenacular or Sertoli cells. These cells have a large function. They're attached tightly to that membrane, and they have tight junctions between each other, and these sperm develop and are nourished by, and their development's stimulated by these sustenacular cells. The sustenacular cells do several things. First of all, they provide the nourishment, the materials that these developing cells need for growth and development. They also maintain a blood-testis barrier because we're going to start putting proteins in their membrane that might trigger an immune response. So we have to have tight junctions, and we have a blood-testis barrier. It's not for the whole testis, it's right here between these two cells. Okay? Next, they secrete inhibin. Inhibin regulates the rate of sperm formation. You don't want to go too fast and you produce a lot of sperm that are never used, or too slow, you don't produce enough sperm for fertilization. Okay? So inhibin is very important in regulating the rate of sperm formation. They also secrete androgen binding protein. The androgen we're talking about is testosterone. Where is the testosterone produced?
Adrenal gland.
There's a small amount produced there.
Yeah,
We're
it's all
this
the-
is
It is the.
in the interstitial cells of the testes. Right there. Everybody that? So that is where most of the testosterone in the male body is produced. And that testosterone is necessary in high concentrations to get these sperm to go through the process. So they have a binding protein that sequesters. Remember, testosterone is a steroid hormone, so it's going to go right through the cell, and it's going to go from here right through into the sustenacular cells, and they are going to bind it and keep a high concentration in this area. Okay? They have an androgen binding protein that concentrates any testosterone that comes. It doesn't leave, and it's there to stimulate those cells to divide and develop. Okay? Absolutely required. And they also secrete the fluid to have these sperm go moving on through the male duct system. So those cells are quite important. Someone in my last class was making a joke. Even at this age, males need a nurse. Okay. Bad joke. Okay, so everybody see the arrangement here? We're now going to take spermatogonium, one germline cell, and see it produce sperm. Now, spermatogenesis begins out here, ends down here when we have a free morphologically looking sperm. So the first step is the spermatogonium. That is a stem cell, a germ cell, okay? And it's going to divide, and remember there's lots of seminiferous tubules and lots of spermatogonia. So they divide and produce a primary spermatocyte. It's diploid. Yeah, it has 46 in there instead of 2 in. So it has 46 chromosomes, and that primary spermatocyte is going to go through meiosis. And they look like normal cells. So this primary spermatocyte is going to, it produces 2 cells. One stays a spermatogonia, the other becomes usually a daughter cell. And the daughter cell is going to go through meiosis 1 and produce 2 haploid secondary spermatocytes. Then each of those is going to go through meiosis 2 and produce 4 haploid spermatids. They're called spermatids because they're going to form sperm, but they're not sperm. Okay? They're normal morphologically looking round cells. Okay, at that point. These are haploid, and those now have to go through a second part of spermatogenesis called spermiogenesis. In spermiogenesis, a haploid spermatid is going to go through morphological changes to form a functional sperm. So all of this from the spermatogonium to sperm is called spermatogenesis. This last part from here to there is called spermiogenesis. It is part of spermatogenesis. The last part of spermatogenesis, taking these haploid spermatids and forming sperm or spermatozoa. Okay? And for the male, one primary spermatocyte produces 4 functional sperm. That is true for the male, but not for the female. We'll see that shortly. Any questions on spermatogenesis? And the book talks about the morphology of the sperm, and you can go and look at that. That's a pretty simple section. Now, semen, the semen only contains 5% of its volume is actually sperm coming down from the ductus deferens. Okay? Only 5% of the volume of semen is actually sperm. The other 95% is male accessory gland secretions. So a volume of semen, only a small fraction is actually sperm, concentrated sperm, coming from the ductus deferens. So what glands produce the secretions that make up most of the volume of semen? First of all, the seminal vesicles, they're sometimes called seminal glands. They're right next to the ampulla and enlarged end of the ductus deferens, and they join together to form the ejaculatory duct that goes through the prostate. That gland produces 60% of the volume of semen. So most of the volume of semen comes from those seminal vesicles or seminal glands. The solution is alkaline. Okay? The secretion of that is alkaline to neutralize acids that are present in the female reproductive tract. The female reproductive tract is acidic to prevent the growth of bacteria. Well, these sperm are pretty fragile. They can't exist and survive in a acidic environment. So semen contains a lot of bicarbonate ions and other ions that make it alkaline. In that secretion coming from the seminal vesicles, there's fructose and citric acid. That is nutrients for the sperm. They need energy, easily metabolizable molecules, so they can generate some ATP to move their flagella and swim. Okay? So this is food for the sperm. That only gets them a small distance. The rest of the way it's provided nutrients by the wall of the female reproductive tract. They secrete other molecules that can be used. This gets them started. There's prostaglandins, in semen put in there by the seminal vesicles that help stimulate smooth muscle contractions to help the sperm move in the direction they're going. And finally, there are clotting proteins in semen to make a clot. To make a clot. So when it's deposited, it doesn't immediately run out. It stays there, keeps the sperm there, and it slowly dissolves away before it gives time for the sperm to leave on their trip. Next, the prostate gland, as you know, encircles the urethra. The ejaculatory duct goes through there, and its secretion makes up about 30% of the volume of semen. It's alkaline also to neutralize acids in the female reproductive tract. It produces citric acid, okay, and clotting factors that work with these clotting proteins, of course, to clot. It forms an actual clot. And proteolytic enzymes to break the clot down slowly so it can release the sperm, and they stay close to where they were deposited and have a good chance of swimming the distance. Okay? Alkaline. Finally, the bulbourethral glands lie inferior to the prostate gland in the wall, the pelvic floor. The muscles there is located among those muscles. And these glands produce a very small secretion. It's only about 5% of the volume of semen. It is an alkaline mucus. Now, because of its location, so you've got the bulbourethral glands here, you've got the prostate gland here, the seminal vesicles here. When semen is produced, that's the first part that goes through the male urethra, and the male urethra is also acidic because urine goes through there, and urine is acidic. So this neutralizes acids in the urethra. So that's everything you needed to know about semen. Alkaline secretion. Every gland has alkaline secretion, and most of the alkalinity is going to be due to bicarbonate ions. Physiology of male reproduction. Male reproduction, of course, is mostly under hormonal control, and it's a very simple system because it's going to produce a steady state level of testosterone. So, as you know, gonadotropin releasing hormone from the hypothalamus stimulates the anterior pituitary to release luteinizing hormone, LH, and follicle stimulating hormone, named after their effects in female hormone. So these two are released. LH stimulates the interstitial cells of the testes to produce testosterone. Testosterone has negative feedback on GnRH and the release of LH and FSH. That causes a male to have very constant levels of testosterone. Testosterone has several effects in a male. That'll be on the next slide in more detail. Okay? But this produces a steady level of testosterone in the male. Now, we'll look at this before we go any further. If a male injects anabolic steroids, they're injecting testosterone. What happens to the testosterone levels? Higher than normal. Yes, you get changes to male behavior. Roid rage is what some people call it. Okay? But what is that going to do to the level of GnRH?
It's going to stop the production.
It's going to be lower than normal. What about the level of LH and FSH? It would be way down. So a person who is injecting testosterone, it's easy to tell if they're injecting testosterone because that's low and those are low. They just do a blood test on these athletes and they say, oh, you've been using anabolic steroids, and there's no way around it because you just look at the levels. Do different males have different levels of testosterone? Yes. That's natural differences, but if you look at those individuals that have more testosterone, they have reasonable levels of the other two. Ashton?
So is like taking testosterone, like pills, like replacement, is that the same as anabolic steroids? Does it affect
No.
with that?
Testosterone is not absorbed well. It's not, it doesn't enter in as testosterone. So you have to inject testosterone, from what I understand, it has to be injected. Pardon? Did you say something, Corey? Oh, okay. So it has to be injected. Can you take other supplements that might stimulate the production of it? Yes. But if it does, it's gonna work with this mechanism, so you'll have maybe a higher level of this, but you'll still have natural levels of those too. So there are supplements that have a small effect on increasing testosterone levels, but it leaves these levels unchanged. I mean, normal, they're normal. Okay, now, LH is the one that stimulates the interstitial cells to release the testosterone. Testosterone has effects in the male. FSH has no follicles to stimulate, but there are sustenacular Sertoli cells and spermatocytes that need nourishment. So this increases the rate of sperm production. This hormone causes and leads to sperm production. This testosterone is required for it. And of course, the result of this is going to be sperm. Now, if an individual is injecting testosterone, what happens here?
Who's that?
There's plenty of testosterone, but what are we missing? FSH. So not only will they have high levels of testosterone, low levels of those, and low levels of those, they'd have a low sperm count. Okay? So, that's the hormonal, oh, forgot one other thing. As I look back, remember, this is negative feedback, these are stimulatory. The sustenacular cells release inhibin to keep the rate of sperm production constant. If it slows down, they release less inhibin, and you get more stimulation. I'll get to you in a second. And if this is going too fast, they release more inhibin, and it slows it down. So there's a balance, this negative feedback loop. This negative feedback loop regulates sperm formation. This negative feedback regulates the level of
The
testosterone.
Sertoli cell only regulates the FSH.
It only regulates FSH
No
secretion,
LH.
not LH, doesn't touch it. Your question?
How could, lower counts of sperm due to steroids be permanent?
If you take these, no, your sperm counts will be low. If you are injecting testosterone, your sperm counts will be low.
How could that
If
be
you,
permanent?
huh?
How could that be permanent when you stop injecting?
When you stop, this system, assuming it hasn't been destroyed anyway, lower testosterone is gonna cause the secretion of this, cause the secretion of this, and you'll rev this up again. You'll restart spermatogenesis, it'll go faster. Now, when you have high levels of testosterone, a little FSH, you're producing a few sperm, but the sperm count's too low. You stop taking the testosterone, natural testosterone will go up, so you'll have natural levels of these, and this will respond. The testes might shrink during that time, don't know. That I don't know. But sperm production should resume. They're just running slow because there's a little FSH, because you got too much testosterone. Any other questions? Testosterone has many effects in a male. We have touched on several of them as we have gone through A&P. Before birth, about 5 weeks after fertilization, the embryo has a female duct system and a male duct system, okay? And at about 5 to 6 weeks, the male embryo begins producing testosterone. It stimulates the development of the male duct system, and it causes the female duct system to deteriorate. Okay? If it's a female embryo at 5 or 6 weeks, there is no testosterone to stimulate the female reproductive system, the male reproductive system, so the female duct system will develop and the male won't. Okay? So born with two duct system, different names, different origins, and one develops for the male, one develops for the female. That testosterone also stimulates the development of the male brain. It's wired differently. If there's no testosterone, the default is the female brain. It's wired differently than the male. The cause of the difference is testosterone. They're wired differently, they have different responses. It's all based on testosterone back at that time. You start development of the male brain, okay? And also external genitalia will also develop as a result of the testosterone. Now, the amount of testosterone and the type of testosterone that's there is very weak, but it's enough to cause all of those changes. At puberty, it's going to stimulate the development of the male sexual characteristics. And then after puberty, it stimulates, because the levels are low, really, they're low, lower yet, and now they get very high. It's going to stimulate male sexual behavior, spermatogenesis, the male sex drive, protein synthesis. It maintains the male secondary sexual characteristics. It also stimulates red blood cell production, and you end up with higher hematocrit. So there are a lot of changes that occur in a male because of testosterone. And if you ever asked why is the difference between a male and a female, almost always the answer is testosterone. Without testosterone, the embryo will develop as a female. For example, if there's a gene that's missing, that does not allow the embryo to produce testosterone, it's still male with a Y chromosome, but it develops as a female. Okay, this kind of story right here is always interesting to look at and see how development occurs and how you can take the same genome with just a few genes really different and develop into a male or a female, which are quite different. Okay, the female reproductive system. This one is a little more complex in structure and function. It's got to do more things. So as we go through here in the female reproductive system, you're going to see it's pretty simple in the male and it gets pretty complex in the female. So here we go. Let's talk about the production of eggs. Eggs are called ova, okay? Or singular would be ovum. The process to produce eggs is oogenesis. Some people like to try and say ooh. No, it's oo if you're pronouncing it right. So oogenesis is comparable to spermatogenesis. Spermatogenesis begins, and 75 days later or so, you have a mature sperm. Okay, so it's fairly straightforward. Oogenesis takes a long time, and very, very few eggs, oogonia, actually complete meiosis. So oogenesis, in oogenesis, when we're developing the four products of meiosis, we do not divide the cytoplasm equally. So in the male, we took one primary spermatocyte, made two equal sized secondary spermatocytes. In the female, putting all the eggs, all the cytoplasm in one, and the other is called a polar body. So instead of producing in a female two secondary spermatocytes, we produce one secondary spermatocyte, which is much, much larger than the polar body. Think of the polar body as a convenient receptacle for chromosomes you're not going to use anymore. They just stick them in there. It doesn't ever usually divide. It has so little cytoplasm, hardly any organelles, and it's just a small, tiny cell. And you'll see that right here. Well, they're not drawn to actual size. Okay. So at the end, if it goes all the way, which is rare in a female, if it goes all the way through oogenesis, then you're going to end up with two or three small polar bodies and one huge ova. An ovum is a huge cell. If you take an ovocyte that has gone all the way through and has been ovulated, you can see that against a black background. Whereas you got high power, you have trouble seeing it on microscope. The sperm, it's the big difference in the sizes. Now, before birth, oogonia produce a large number of primary oocytes. So before birth forms millions of primary oocytes. They begin meiosis and all of them stop in prophase 1. This is, again, before birth. So the ovaries of a newborn female contain millions of primary oocytes. Every one of them has begun meiosis. Remember, meiosis begins by contracting, condensing the chromosomes so you can see them. That's called prophase. That's where every one of them stop. So the ovaries are full of a large number of cells, oocytes, that are primary oocytes that are in prophase 1. They begin the first division. They just condense the chromosomes and they stay like that. Okay? And now, between birth and puberty, a lot of them deteriorate, degenerate, and are dead and gone. Okay? After puberty, every month, a few of those primary oocytes will develop further. Say 24 begin development every uterine cycle. Okay? 24 begin. And then of that, now it may take more than one uterine cycle, one cycle before they're mature, but out of that group, only one is going to become mature. Okay? Just before ovulation occurs, that primary oocyte arrested in prophase 1 will finish meiosis 1, kick out a polar body, first polar body, and then begin meiosis 2 and stop at metaphase of meiosis 2. So we started and stopped again. Everybody see that? What is ovulated is a secondary oocyte arrested in metaphase 2. The chromosomes are all lined up on the metaphase plate. That oocyte that is ovulated once a month, usually just one every cycle, it will only complete this meiosis 2 that it started if it's fertilized. If it's not fertilized, it stops there and never goes any further. If it's fertilized, it'll finish meiosis 2, fertilization, and then fuse the nuclei, kick out a polar body, and you have a new zygote. Okay? So, oogenesis is quite different from spermatogenesis, where all these cells are all going through the same process, and each primary spermatocyte produces 4 sperm.
You said before birth it stops at prophase. Prophase 1.
Yep, prophase 1. That's prophase of meiosis 1. It's usually called prophase 1. And then the one that is ovulated, just before it's ovulated, it finishes meiosis 1 and begins meiosis 2 and stops in metaphase 2. It will not complete meiosis unless it is fertilized. Yes?
Alright, back to my question. On the diagram it says early and middle prophase 1. So after middle prophase 1?
Just learn it as prophase 1, early and middle.
I just
We're not
wanted-
gonna split hairs here. If you know it stops in prophase 1, we're good. Okay? Metaphase is a very simple phase. That one is more complex. And you can see here, this is before birth, and it doesn't say the millions there. It says decreases to 300,000 after birth. And then once a month, one finishes, ovulated, that's the secondary oocyte, it's arrested in metaphase 2, and if it's fertilized, it will complete meiosis. If it's fertilized, completes meiosis, kicks out the second cord body, and then becomes a zygote. Okay, let's look over at the other side. So this is the development of the oocyte. The oocyte is part of what? A follicle. A follicle is an oocyte with surrounding cells. So, oocytes produced in, that are produced here are called primordial follicles. They're surrounded by a layer of cells that are squamous, okay? So they're called granulosa cells, but the primary follicles have squamous cells around them. Not much is happening. The oocyte is just being nourished by those follicle cells. When they become primary follicles, those squamous cells become cuboidal. Now, a squamous cell is just covering, surrounding. A cuboidal cell has more cytoplasm, and they begin nourishing the oocyte. It grows, they form a zona pellucida, that clear layer that we'll talk about in chapter 29, okay? And additional cell layers will be put on it, so it has a little more than one. So that's still just a primary follicle. Primordial, squamous, cuboidal, and now we're gonna put another layer or two maybe on it. Next, it goes and it's going to continue to have cell layers. When it starts to become a secondary oocyte, we're gonna have fluid form between cells. The cells absorb and secrete the fluid that's going to form this antrum in here. So we start to see fluid between some of the cells. At the same time, we're going to produce a layer of squamous cells around the outside that's called the theca. The theca and the granulosa cells work together to produce estrogen. This follicle, these granulosa cells and the thecal cells produce estrogen. They've got to work together. One cell does one part, one cell does the other part. Okay? At the point we get a theca, right here with a secondary follicle, we're beginning to get a production of huge amounts of estrogen. Okay? And that secondary oocyte in here, once it becomes a mature or a tertiary or a Graafian follicle, this cell is going to be finishing meiosis 1. Okay? And we're going to end up in a mature or tertiary follicle. You're going to have a huge number of cells around the oocyte. The oocyte gets bigger. It's going to give off one polar body, which you can't see. And there's a huge antrum. Sometimes it projects out and is actually on kind of a stalk, if you see it just right. Okay? At ovulation, we already had this begin. Once this tertiary is formed, the cell, it takes a while, goes through and completes meiosis 1, starts meiosis 2, kicks out a polar body. It's in one of these other cells around it. And then when it's ovulated, there are adjacent cells attached to it that go with it. So this is ovulated into the pelvic cavity. Right there on top of it is the fimbriae of the uterine tube, and they almost always go up in there. And just as they enter is about where they're usually fertilized. And the cells, the granulosa cells, they're called cumulus cells right here in a tertiary follicle. Those cells, which are basically granulosa cells or follicle cells, attached to it are called the cumulus cells, and this is called the corona radiata oftentimes. Okay, that's what's ovulated. What remains inside the ovary, because that's the wall of the ovary right there. This is ejected, and that stays in the wall of the ovary, then seals back up, and the remaining cells become the corpus luteum, and that is involved in secreting estrogen and progesterone. So that's what happens to the follicle. This describes what's happening to the oocyte. This describes what's happening to the follicle. Covered that, I covered that. Okay, physiology of the female reproductive system. The female reproductive system is under neural and hormonal control, just like the male. So, this is a chart of several things. We have days across the bottom, and we're beginning with menstruation right here at the beginning of it as day 0. Okay? And after 14 days after the beginning of menstruation is about when ovulation occurs, give or take a couple of days. We're using a 28-day cycle, although the cycles vary in their length. Okay? So this is menstruation, lasts 4 or 5 days. Then you begin what's called for the uterus. This is the uterus panel. You begins the proliferative phase because this is replacing the functional zone that was lost. Okay? And glands grows in thickness. And these glands, what do you think glands are for? To secrete something. So implantation will be over here, and that embryo that is able to get nutrients from the secretions of these endometrial glands. That's what they're for. Now, this is called the proliferative phase if we're talking about the uterus. This time for the ovary, it's called the follicular phase. Okay? So if you're referring to the uterus, what are we talking about? Proliferation. If we're referring to the follicles in the ovary, we're talking about developing follicles. So that's what's happening here. So there are 2 names for this phase. At day 14, approximately, we have ovulation. Then this phase is called the secretory phase if we're talking about the uterus, because they're secreting to nourish if there's any fertilized, if there's an embryo, it's what's implanted. The actual fertilization occurs down the uterine tubules, but we have to be secreting when it comes in around this time, day 5 or 6 after fertilization. What's happening up here at the ovary? We have the corpus luteum actually dominating, and that's what's happening here, is it's secreting estrogen and progesterone. So this is called the secretory phase, referring to the uterus. It's called the luteal phase if you're referring to the ovary. Okay. So in sequence, we have the following panels. This is what's happening to the endometrium. This is what's happening to the ovary. Here is FSH, LH and FSH. And this is estrogen, and that's progesterone. Did I mention that female hormones tend to go up and down? Yes, they do. Now, not mentioned in this, remember, we're going to have the hypothalamus release GnRH. They're not showing you GnRH. GnRH is going to stimulate in the male both FSH and LH release. Even in the male, GnRH is released in pulses, but it's a fairly steady state, so it gets a constant levels of LH and FSH. In the female, the rate of release, the frequency of those pulses changes. This isn't mentioned in the book. And it can preferentially secrete LH or FSH. Okay? So in the female, GnRH and these 2 have a different relationship. GnRH can stimulate one more than the other. And we're going to see that occur as it goes through. Okay. On day 1, menstruation begins, the endometrium is sloughed off, and we now have an increase in FSH. And what is FSH again? Follicle stimulating hormone, which stimulates at this time. It's increased from a basal level, and we now get a stimulation of beginning the development of several follicles. They're going to be primary, and then they're going to begin to develop. Anyone that begins 1 month probably isn't going to complete that month, okay? It may take several cycles before they're mature. Most that start don't become ovulated. Most that start are not ovulated. Maybe 1 out of 15 or 20 that begin are actually ovulated. Okay. After menstruation and beginning to develop follicles, we're going to have the proliferative phase. On day 14, ovulation occurs, GnRH is going to stimulate, where are we? Okay. GnRH, it will stimulate the release of LH and FSH here, and they're going to remain high during this time, and together they stimulate follicle growth. During this time here, during this period here, we're going to be having follicles grow. They have high levels of both FSH and LH, okay? FSH starts them developing. LH and FSH continue their development, cause them to continue. I already covered that. FSH and LH are then going to stimulate the follicle cells to begin to release estrogen. As the number of layers of cells increase and it becomes a secondary oocyte, you get the production of estrogen. The estrogen is going to stimulate an increase in LH. FSH a little, but LH a lot. Estrogen is going to stimulate the release of GnRH at a certain pulse frequency that favors LH. They both go up, but which one goes up the most? LH. And LH is what triggers the completion of meiosis and ovulation. So the pulse here is going to cause ovulation. The developing follicles are going to secrete inhibin, which is going to cause FSH to go down. Okay? They're secreting inhibin and it goes down. And it goes down to a fairly low level compared to what it was over here. The levels are not too revealing there. I had the LH surge. Now, the remaining cells are going to form the corpus luteum, and that's going to secrete estrogen, but mostly progesterone. The progesterone, the endometrial cells have put progesterone receptors on them, and that progesterone keeps them there and secreting. Okay? If an embryo is going to come in, it's going to come in around day 20 and begin to implant. That embryo has just a few days before this layer that it's implanting into is going to be released. And the embryo has to produce human chorionic gonadotropin that causes the corpus luteum to continue its progesterone level. Without HCG, progesterone from the corpus luteum is going to decline. With HCG, it's going to maintain high. That will keep the functional layer of the endometrium intact. If the oocyte is fertilized, remember, it's going to implant, produce HCG, keep the corpus luteum producing progesterone that maintains where it's living at that time. Later on, around 2 months after pregnancy, this will begin to generate as the placenta produces the progesterone. So it's got to go for 2 months in the ovary. That one group of cells producing progesterone to maintain that. If fertilization does not occur, what happens is estrogen will begin to decline, progesterone will begin to decline. The declining progesterone itself causes this. The declining estrogen causes the production of more LH and FSH. They go up, and the FSH stimulates the next ovarian cycle. The declining estrogen stimulates GnRH, FSH, and LH, this increase right there, that begins the next ovarian cycle. Declining progesterone causes menstruation. That is the cycle and all of the major events correlated. If you look at the control of the female reproductive system, this is what it looks like. This is a diagram out of your book. So we have off the screen, unfortunately. Let me reduce that so we can see it. Just a second. I can reduce the size of it so it doesn't go all the way to the top and get lost. Now we got the top. Okay. Remember, the hypothalamus not shown is, as far as the labeling it, GnRH is gonna stimulate FSH and LH. FSH is gonna cause follicles to develop. LH is gonna cause them to further go. The follicles, when they develop, begin to secrete estrogen. The estrogen is going to have a positive feedback on FSH and LH. Why are we doing this? And that feedback is more strong on LH. Why are we having that? Why does estrogen have positive feedback and stimulate LH? You know? So we can have ovulation. LH is gonna work with FSH to get the follicles to develop and mature, but a surge of this causes ovulation and causes what's left of the follicle to form the corpus luteum. Okay? And the corpus luteum is gonna produce estrogen and progesterone. That has negative feedback on LH and FSH and the release of GnRH. After this is gone, we have none of that, and now we have stimulation of this to produce more FSH and LH. The estrogen is going to cause the uterine epithelium to proliferate. Now, this whole side is in the follicular phase, this is the luteal phase. And estrogen and progesterone maintain the endometrium layer. Not shown here is inhibin, which has a negative effect once they're developing, a negative effect on LH. But that's only late in the cycle. A little more complex, isn't it? Yeah. So we have no questions. Do we have time for questions? Yes, we have time for questions. Let's see what I got for questions. I can put them in. In the interest of time, I have kind of hidden them so they don't show up, but I can go through the questions. First question, what effect would low levels of FSH have on sperm production? It would be decreased, low. Okay? Low levels of testosterone and low levels of sperm production. John contracted testicular cancer as a teenager and surgery removed one of his testes. Will John be able to father children and retain male secondary sexual characteristics? Yes or no? You are correct. And that was a high school friend of mine, and I've met his son Joel. Okay. So, yes. Well, yeah. Okay, high school friend. What changes would you expect to observe in the ovarian cycle if the LH surge did not occur? No ovulation, anything else? Estrogen would stay high because you're not forming the corpus luteum. You don't have the LH to tell them to shift from estrogen production to estrogen and progesterone. The formation of the corpus luteum wouldn't occur either. Okay? So, the LH surge is very important. It triggers ovulation and the next phase. What effect would a blockage of progesterone receptors in the uterus have on the endometrium? It wouldn't what? Now, it's already developed. Estrogen stimulates the development and it gets thicker. Proliferation occurs. If there's no progesterone receptors, what happens next?
It couldn't, the embryo couldn't develop.
Well, the embryo couldn't develop, but more than that.
Can implant.
Hmm?
If there is no proliferative phase, so there is no implantation.
Implantation wouldn't occur because as soon as the corpus luteum is supposed to be producing progesterone, it is, but there's no progesterone receptors, and it would begin menstruation shortly after ovulation. So implantation couldn't occur because we're gonna be in an early menstruation. You need the progesterone onto the progesterone receptors to keep that tissue intact. Would inhibit development, it would make the uterus unprepared for implantation. It would also have an early menstruation. What event in the uterine cycle occurs when the levels of estrogen and progesterone decline? What's gonna happen next? Yeah, menses. Okay. And that's all the questions I had. Now, some of you came in late and do not have your lab practical. I have that here. So if you don't have your lab practical or your test report, please come up here. I will give them to you. After I finish and leave here, I'm going up to my office, you can come look at exam 4. Bring your test report, you can look at exam 4 if you have notes. Okay?